This was a tricky question. What you needed to do for this one was use this formula to work out the volume of the two circles. So to work out the volume of the polar body and to work out the volume of the nucleus within the polar body, so work them out separately, find the difference, which would be 409, and times that answer by the number of mitochondria per micrometer cubed. So times that answer by 0.08. Okay, quite a few people got the wrong end of the stick with this question. So mitochondria are found in the cytoplasm of the egg. The genes for healthy mitochondria are not found in the nucleus. Okay, the mitochondria have their own set of circular DNA. So to prevent the mitochondrial disease, so using this technique, you need the nucleus from the mother. So that'll be the first mark. So you've got the genes in the nucleus from the mother. So that's what the egg has, because you've taken it and you put it into the egg cell from the polar body. And then that egg cell that's got from a healthy, that's got healthy mitochondria in it, we have lots of normal mitochondria. So there won't be any or very few faulty mitochondria in the egg. So there's another classic example where people are just writing what they know but not explaining it, not applying the knowledge. So if you've got faulty mitochondria, what's going to happen straight away? You should be saying there's less ATP or not enough ATP is produced. Then you need to think about, well, how can ATP relate to, they might be important for enzyme catalyzed reactions. So ATP can do two things. So ATP could provide energy for those reactions to take place. So the hydrolysis of ATP provides energy. Or the ATP could phosphorylate the enzymes, so making them more reactive. Or because they lower the activation energy of the reaction to happen. Okay, so this question is about the cooperative binding of oxygen to hemoglobin. So it's the idea that when the first oxygen binds, it changes the tertiary or the quaternary structure of hemoglobin. So when the first oxygen binds, it's difficult for that first oxygen to bind. Once it does, it changes the tertiary structure of hemoglobin. Therefore, what does it do? It uncovers further heme group binding sites, so the other three oxygens can bind. For the next maths question, it asks you to calculate the number of red blood cells per millimetre cubed in the original blood sample. So some important things that you need to get from this is that the blood sample has been diluted by 200 times, and we've got the length of each side of the square, because we need per millimetre cubed in the answer. So we need to work out the number of millimetres cubed that this box is. And we've also got the depth. So volume is the width times the length times by the depth, because it's a cuboid. So the calculation would be, there are 7 counted, so 7, you'd count 7 yeast cells, because remember you don't count these two here. So 7 times by 200, and that will give you the number of cells in the original sample. Okay. Then what you'd have to do is work out the volume of one square. So the volume of one square would be 0.05 times by 0.05 times by the 0.1, which is the depth. So that is 0.00025. So then what we need is the number of cells per millimetre cubed. This is the number of cells, this is the millimetre cubed, so it's that divided by that. So it's 1400 divided by 0.00025, which gives you 5.6 times 10 to the 6. This type of questions come up a few times, usually to bacteria, counting bacteria, linking to require practical 6. This time it's with white blood cells. So why do you only need to dilute a certain number of times? Well, the idea is if you use the pure sample, there'd be too many to count. You need to dilute it enough until there's the correct amount to count. So why only 20 times? Well, if you diluted it further, there'd be too few white blood cells to count. It wouldn't be enough to count. So that's why you only need to do it like 20 times. Just make sure if it says suggest why, you give a point and an explanation. So the idea is there are too few white blood cells, I suggest. Are there fewer white blood cells, I suggest, and then why? So you don't need to dilute it further.